Okay, so tonight we're going to be talking about different kinds of dominance. We've already learned sort of plain old dominant versus recessive, brown eyes, blue eyes, yellow peas, green peas, tall, short, pea plants, etc. And so tonight we're going to be clouding that picture a little bit, talking about some of the exceptions and different things that can happen. Obviously, the traits you observe in the world are not just, binary, it's A or B, and nothing in between. So our first up is incomplete dominance. And this is pretty much what it sounds like. Neither allele for a trait is completely dominant over another allele, and so the resulting phenotype is a blend of both alleles. For example, snapdragons can be either red or white, and when a snapdragon possesses both alleles, not surprisingly, it'll be pink. Um, so here we have the red snapdragon, the white snapdragon. These are our parents. When they make babies, they are all pink. And so what you've noticed is that the pink offspring don't look like their parents at all. I'm sure all of us know people who don't look like their parents. We've got red, white, and when we cross them, each offspring is going to have an R and a W allele, so each offspring is going to be pink. Alright, so then what happens when we take two pink snapdragons and we mate them? These offspring will express the phenotypes of the parents and their grandparents. So we've got pink, pink, each with an R and a W. And so then they create a RR, a WW, and two RWs. So they'll have a quarter of their children red, a quarter white, and half pink. So you can take a minute, calculate the genotypic and phenotypic ratios for these. Alright. So next up, we have another example of incomplete dominance. Another color blending one. If you take a black sheep and a white sheep, you get a gray sheep. I want you to take a moment and complete the following crosses. So you take the black sheep, mate it with the white sheep, and you'll get a gray sheep. And so black is noted as big B, white is big W, big W. And so I want you to do that cross, and then I also want you to cross the gray sheep with the gray sheep, so they both have one of each allele. And then take a minute and calculate the phenotypic and genotypic ratios for both of these. Alright. So next we have codominance. Both, in this case, is very similar to incomplete dominance, but in this case both alleles are expressed. This creates a combination rather than a blending, and you'll see what I mean in a sec. So for example, if you take a white chicken and a black chicken, unlike the sheep, they don't make a gray chicken. They make a speckled chicken. So we've got our white chicken flying in, our black chicken, and they make little babies with EW genotypes. And they are all this lovely speckled phenotype. Okay, so then we have what happens when you take a speckled chicken, mate it to another speckled chicken, and once again we get the parents right here, speckled chicken phenotypes, and you get the grandparents, the black chicken and the white chicken. So take a minute and calculate the genotypic and phenotypic ratios for this cross. If it looks similar to incomplete dominance, that's not surprising. It is similar, it's just slightly different in terms of blending versus combination. Okay. So next up is sex-linked traits. It's exciting, sex, but most sex-linked traits actually have nothing to do with the sexual characteristics. So these are usually traits that are coded for by genes on the X or Y chromosomes. They're just usually like regular old body traits. So hemophilia is something that can be X-linked. There's also color blindness, which is what we talked about. And so what we've noticed actually is that recessive sex-linked diseases often affect men a lot more than women. So we're going to find out why. So the first thing we're going to talk about is color blindness. If you don't know what it is, take a look at this picture. If you can't see a number, you might be colorblind. Number is 56, so test yourself. Alright, so we have, as you know, women, 2 X chromosomes. Men, 1 X chromosome, 1 Y chromosome. That's the Y chromosome, super tiny. The X chromosome is much bigger than the Y chromosome. It codes for all sorts of traits, even those unrelated to biological gender. Alright. So color blindness is coded for by a gene on the X chromosome. Color blindness is a recessive allele and color vision is dominant. This makes sense evolutionarily. Color, helpful, seeing things, finding prey, taking shelter, etc. So what happens when a woman has one X chromosome with a colorblind allele and one normal X chromosome? As we can see, we have the colorblind allele, the X chromosome, and she's going to have normal vision because she's got that one good allele and one, colorblind allele. But the good allele is dominant, and so she'll have normal color vision. So what happens when a woman has two X chromosomes with a colorblind allele? It's a little bit different. So now she doesn't have any choice. She only has the colorblind alleles, so she's going to be colorblind. Alright, so what happens when a man has just one X chromosome with a colorblind allele? Remember, he doesn't have another X chromosome. So he has the colorblind allele, and then this, sad little Y chromosome over here. And he's going to be colorblind because he just has that one colorblind allele and no other alleles. So pretty much any disease or any trait that is coded for on an X chromosome alone will affect men a lot more than women, because women will have 50-50 chance of getting it covered up by a good chromosome, a good X chromosome, and men just have this Y chromosome, which doesn't really line up very easily. Alright, so now we're going to try some crosses with color blindness. Because we have the X and the Y chromosomes and they're different, we actually mark it a little bit different. So we'll mark color blindness as a C, a little C superscript, next to the X. And so that's going to be colorblind. Just a regular old X is our normal X chromosome. And then Y is Y because it doesn't carry any genes related to color vision. So we have our X and Y sperm from the father, and our colorblind X's from the mother. In this case, we're crossing a colorblind woman with a normal vision man. And so what we find out is that their daughters are going to have normal vision, but both of them are going to be what's called carriers. They're going to have this recessive allele. It won't be expressed. And so anytime, actually, that you have a recessive allele that's not expressed, that's considered a carrier. So you have brown eyes, but you have a blue eye allele that you have, but it's not being expressed, you're a carrier for blue eyes. Normally we talk about this in relation to disease because we think of like carriers, carriers for disease can spread disease. In any case, we've got normal vision daughters and colorblind sons. Alright, so take a minute, calculate the genotypic ratios, phenotypic ratios, the following cross. Alright, and that's it. So thank you so much for watching, and see you tomorrow.